Hello and welcome to the Interagency Modeling and Analysis Group webinar series, and I'm very happy to introduce the host of this webinar today is the Cell to Macroscale Working Group, and the working group leads are Dr. Qinglong Lin and Dr. Ed Sander, and I will turn it over to them. Thank you.
Thanks, Grace. I just want to say that the working group aims to discuss and share numerical methods that facilitate integration of subcellular and cellular scale to macroscale in the human body and identify associated multiscale modeling issues from the numerical standpoint. And so as part of that mission, we have a series of webinars presented throughout the year. And today's speaker is Dr. Hans J. Johnson. He is an assistant professor in the Department of Psychiatry at the University of Iowa Hospitals and Clinics. He received his PhD in electrical and computer engineering from the University of Iowa in 2002. He is involved in several relevant projects for the IMAG sort of level of interests. And his primary research interest involves accelerating research discovery through the efficient analysis of large-scale heterogeneous multi-site data collections using modern high-performance computing resources. Some other just tidbits of his extensive bio include that since 2010 he's been the president of Insight Software Consortium, which is a nonprofit educational consortium dedicated to promoting and maintaining open source freely available software for medical image analysis. And as I said, there are many other software related, multi-processor, high performance computing related applications that he's deeply involved in. Today, he's going to talk to us about managing complexities of collecting, curating, and performing analysis of big data. And so without further ado, I'd like to present Dr. Hans Johnson.
Thank you very much. I am going to talk today about a couple of big projects that we work on, mostly related to medical imaging data analysis and all of the support structures and other data that go along with those types of studies. Multi-site and longitudinal are two key words that I'll touch on throughout this presentation, and some of the complexities and difficulties that we run into when dealing with multi-site and longitudinal data. So today's buzzword is big data, of course. Everybody hears about big data all the time, and so I thought I'd just start off here with a quick description of what big data is, right from Wikipedia. And I think that this underlying section, the challenges include the capture, curation, storage, search, sharing, transfer, analysis, and visualization of these very large data sets is spot on to the problems that we run into in our neuroimaging and clinical endeavors. This picture is a graph of the data storage in our lab here in the psychiatry department from '93 up through about 2009. And as you can see, our growth has happened quite rapidly. Around 2002, we started collecting and generating a lot more data. Our growth curve drastically changed from before 2002. So in the last decade, we're starting to apply new algorithms and new methods like this one shown here for using high-dimensional nonlinear warping. And this experiment it caused a huge jump in the amount of data storage that we had. So as we take new cutting-edge technologies and apply them to the data sets that we have previously collected longitudinally, we often run into these huge jumps in the data storage and data processing requirements that we want to address. And throughout the presentation today, I'm going to talk about how we address these types of jumps that are occurring as we bring in novel, new, cutting-edge analysis methods that really challenge our ability to simply process data and present it back to the user community. So I don't know how many of you are familiar with Ron Kikinis. He's the head of the National Alliance of Medical Image Computing, and he has a real focus on creating translational clinical research tools. He wants to create tools and help others create tools that many people will use. So he developed these simple rules for tools that really describe how to take your cutting edge tool that your research assistant or that you have created and get it to the point where it can actually impact translational clinical research for other people. So his first rule is you make it, I break it. Basically meaning that there are many, many tools out there that are fabulous, and they work on one data set in one lab on one person's laptop. And he wants to make sure that those tools work on the data set that they were originally designed for, but also on other people's data sets. So that gets into a second rule. Your tool really doesn't even exist until it works on my laptop with my data. And that gets back to the idea that to make these tools useful and to have high impact of this cutting edge research that we're all trying to do, it's really important that it works on other people's data and other people's laptops if it's going to have a widespread use. He's got a couple about user interfaces. I'm lazy, you don't want to move the mouse too much. You don't want to have more than one simple parameter. Of course, that's not always possible. And this last one is particularly important in the work that we're doing. Is I have ADD, so make your algorithms fast. We're doing neurosurgical planning where we use the computer algorithms to help to guide neurosurgical cases. And in those cases, your skull is actually removed from your head and is sitting in a dish alongside of you. So every second that the algorithm takes in order to process this data in real time is another second that your brain is exposed to the air and complications can occur. So along with Ron's rules, I have a couple of addendums that we try to live for in our lab, which is commit early and commit often. So as we are developing our algorithms, we want to have them tested early. We want to have other people reviewing and identifying changes in our code very quickly in the process. And the second is we have this mantra that says, if it's not tested, it's broken. So each of our tools, we put through a rigorous set of tests and have a set of criteria that I'll go through in just a second for when it can be deployed and used on our data sets. So you may be asking why all this emphasis on the testing part of preparing for big data analysis. Well, many of our studies are actually longitudinal in nature, and they're longitudinal in both the data collection and in the analysis that's done over a span of many, many years. We have a schizophrenia study that's gone on for more than 20 years, and a Huntington's disease study that is in its 14th year, and many other studies that simply have a long time span from when the first data set was collected till the last data set was analyzed. We need to develop the software to analyze the clinical, the genetic, the imaging, and all of the metadata around that, and it can take many years to analyze and collect all that data. So it's important that the integrity of the analysis methods are preserved over time. We need to know when a software product has changed significantly so that it doesn't introduce a bias in the longitudinal analysis. We would hate a bug to be fixed in the 5th year of a project, and by fixing that bug, have an unintentioned bias introduced in our longitudinal analysis. We've seen similar things happen in the past, even when switching from different computer platforms. So exactly the same software, but running on a big Indian machine versus a small Indian machine introduced a bias in the analysis that was generated. So we have to be very careful and keep a close eye on how different compilers, different hardware platforms can affect the actual analysis results. So the BRAINSTools Development, BRAINSTools is our software suite that we develop here. There are more than 200 organizations that depend upon the stability and reliability of that package. It's bundled with NiPype and the BRAINSTools package itself, along with Slicer and many other packages that use components of the BRAINSTools package for brain imaging research throughout the world. And in order to make sure that we don't screw up or jeopardize someone else's research, we need to demonstrate that as we continue to develop and continue to improve this BRAINSTools package, that the results will be the same, or if they're not the same, that we can describe how those results change over time. So we have a complete picture of our development process is organized around a very large regression test suite and a web-based dashboard. So our regression test suite has about 1.2 gigabytes of just reference data that we run every night, and more than 140,000 known analysis results. So every time a change occurs in the code, we run it through the test suite and make sure that the known analysis results are stable. If the known analysis results happen to change, we go and look at them carefully, try to identify why they've changed, and determine whether that change is something we need to alert our user community with, or develop a way to preserve the old results. This dashboard is developed around the CDash Dashboard from the NAMIC toolkit. And it's a dashboard that provides our developers and our end users a means to navigate a series of cross-referenced reports. And these cross-reference reports are generated continuously each night, a couple of times a day, across different hardware platforms, including Mac and Linux and Windows. Those are the three main platforms that our end users use, to ensure that the entire process is stable and that the results of any particular analysis are going to be useful in a further analysis. So this dashboard contains source code management, so it integrates with our Git or CVS or SVN repositories. It does compile time testing, so it will compile the code continuously as new software changes are made. It does runtime regression testing, dynamic memory analysis. It does a code coverage analysis so that we can identify which pieces of our software need to have tests written so we can ensure that they're always working. And it does, we do a static analysis as well to identify potential bugs or potential maintenance issues with the code. I'm going to show you here an integrated view of this development process, this snapshot of the dashboard in simplified terms. But you can see that these reports are generated each night. And with the report each night, they happen on many different computer platforms. A 32-bit Linux, a 64-bit Linux. We also do Mac and Windows builds as well, because we'll notice that not all platforms behave the same. In fact, we've really run into many cases where even the compiler options and the level of optimization that you choose can have pretty significant changes in the way the algorithm performs and the results that are made. And that's important for us to look at so we can go back into the source code and fortify it such that we get the same results or very similar results across all of our different platforms. We would hate to have our Macintosh computers generate one result and our Windows computers generate a drastically different result over time. As you can see in the yellow here, that some of the builds may have warnings or errors, and some of the tests may fail. And you can click on those hyperlinked report items, and it'll drill down and give you an idea of what has changed and what the error is. So for instance, in the warning column here, there's one warning given only on our 64-bit system. And if we click on that, it tells us exactly the line of code that we need to go and review. So in this particular case, we have a variable that's never used, and we can go in and remove that variable to reduce our long-term maintenance costs. In addition, there's a test that failed, and if we click on the failing test, we can see that this is one of the surface tests, and it tells us exactly why this test failed. And if you look at the results in red there, you can see that the results are close, but they're not the same. And this is a very common characteristic of this dashboard system, or of our testing suite, is in many cases, in a multi-threaded, high-performance environment, you cannot guarantee that the results are identical every single time, or at least the cost of guaranteeing identical results is too high. So we wanted to be able to identify what close enough is. And so we have regression tests that identify what a close close number is, so that we can use that throughout and ensure that even though we may get slightly different results on different platforms, we can identify that they're close enough and will produce meaningfully similar results. You can also see here in the lower left that there's updates. So I said that this is continuous building. Every time any developer, and there's at least 30 developers on the BRAINSTools package, anytime any developer makes any commit to the code, that commit is run through the dashboard system. The set of builds on all of our platforms are done, and all of the regression tests are run automatically. And we'll be able to click on those updates and see what has changed in that code. So if there are test failures or build failures, we can drill right down from this report and see what those failures are and address them. And the benefits of this system is that it provides a very efficient self-checking development environment that really has been the key to the success of our Synapse Lab. It's a single code change by one developer automatically triggers the regression test suite on all these different platforms for all these different compiler options and all these different common environments that our end users depend upon. That way we can very quickly and aggressively make changes to the code, knowing that our dashboard system will double check and make sure that, that the results are still meaningful. And it allows our team to work very efficiently because as the team members make changes, and they make them often, as they make changes, they can have confidence that their changes are not affecting different parts of the software suite in negative ways, because the dashboard tells them. And often we can go into the dashboard, look at the red or the yellow areas, click on them, and instantly know how to fix it. We don't have to go searching and pecking through the software code to determine post hoc after 6 months of work have been done what has changed. We see it instantly. If you make a change at 10 o'clock in the morning, by noon all of the regression tests have run, and by 1 o'clock after lunch you can come back and fix your change while it's still fresh in your head and you know exactly what to do. So you may be asking yourself why this is so important. Well, we work with multi-site collaborative research projects, and I'm sure many of you are doing the same. The FBurn project had 11 sites. The NAMIC project is 20 plus sites. The Insight Toolkit, we have more than 1,000 developers that we have to coordinate efforts on, making sure that any one of the changes from those developers doesn't cause a negative impact on other people's software downstream. The TrackHD is a 4-site consortium in Europe. PredictHD has 32 sites, and PredictHD is what I'm going to primarily talk about. And then the NeuroNext initiative. This is an initiative to do clinical trials, where 29 sites have banded together to run many clinical trials in an efficient manner. So I'm going to talk more about the PredictHD project right now, and just a little bit of background. In PredictHD, we're trying to model when we can intervene in the Huntington's disease process. This is an autosomal dominant disease that genetic tests in infancy or at birth can determine whether eventually you will become ill from this disease. And these two plots show what we would like to do. Currently, the red line represents untreated progression of the disease. And currently, the yellow line is what we can do, which is we wait until you have external motor symptoms, basically when you can't control your hand motions, and then at that point we want to intervene. Well, the PredictHD project is trying to collect a lot of data and look at the data before you have these gross external symptoms. We would like to intervene many, many years before the symptom onset and hopefully delay the severity and the onset time of those diseases. In order to do that, we have 32 sites throughout the world, 16 within the US and 16 outside the US in Europe, Canada, and Australia, where each of the sites is collecting neuropsychological, DNA, and MRI data. All these different kinds of data that we collect are then centrally located here at the University of Iowa, curated, cleaned, and then redistributed back to many of the sites for data analysis. And currently we have more than 1,200 subjects in the study that has been going on for 14 years now. So this is a lot of data on many different sites, and this plot shows how we make the most use of our stored resources. In the upper left, we have the Research Center, which is the 32 different sites where we have both expert clinician interviews going on and companions, which are usually wives or brothers or sisters of the participants, doing self-report forms. And sometimes we have participants answering items at home and submitting those. And all that information comes into our central trial management software, our central database, and is organized, curated, and prepared for analysis. In addition to our clinical interviews, we also have more complex data, including derived imaging data, medical imaging data from each of the sites or each of the participants, where we get brain scans in an MRI scanner. Or we also get cerebral spinal fluid and blood and urine that are stored and cataloged and analyzed as well. And all of that information needs to be organized in our central trial management software. Once it's in the central trial management software, we then do a one-time anonymization and make that available through the dbGaP system, where it can be linked to the gene-wide association genetic information for these subjects and made publicly available for analysis by anybody who requests it. So the researchers at the different sites are collecting lots of data. The subjects have about 8 hours of interviews and questionnaires that need to be filled out. And what we did is we did a tablet rollout to each of the sites, where we took 130 computers sent out to each of the sites and configured them so that they can more easily enter the data in an electronic data capture system. Up until about 2007, all of the clinical questionnaires were done with a number 2 pencil and paper, where we would FedEx that data to a central transcription facility, be transcribed, and then put into our database. And that process took about a year from the time that the data was collected until the time became available for analysis. With the electronic data capture, we've been able to accelerate that drastically to just a few weeks from the time the data is collected till the time it's fully curated and ready for analysis. And in important cases, we can do it even much faster than that from any one of these sites. So we had to not only deal with the technical challenges of deploying 130 computers throughout the world, but we also needed to train over 200 people how to use them. And we took advantage of YouTube, of course, and created a set of videos that describe how to use these tablets and do the data entry system. And this electronic data capture really had one huge benefit. I'll get to the benefit in a moment. I forgot about this slide. Our sync protocol manager, because we have over 1,200 subjects at 32 different sites, and each of those different sites have subtle IRB constraints about which forms are acceptable and which forms are not acceptable, and the fact that this is a longitudinal study where different clinical report forms need to be filled out depending upon which visit you're in. For example, your gender is only part of the demographic that's asked at the baseline visit. At subsequent visits, we do not ask what your gender is. We have to have a system that can dynamically change what data is collected from each of the sites. And this is our protocol management tool, where the research assistant or the neurologist enters a few bits of important information, the participant ID and which visit they are, And that brings up dynamically which forms and the order of the forms that need to be collected at that particular visit. And then as they click through each of these forms, it brings up the electronic data capture form that they can use to enter the information quickly. And as they're entering the information in the clinical report forms, they're automatically validated. So for instance, for your current age, if you put in a negative number, it shows up as red and it requests that you change that to a meaningful number. And then each of these forms will change color from red, meaning that they have to be done, to blue, meaning that they're partially done, to green, meaning that they are completed, so that we know that all of the data necessary for this study is quickly entered and sent to our site for central coordination. And the main benefit of this has been that it makes us much more efficient time-wise. So these electronic case report forms have saved approximately 1 hour of participant time in a study that takes about 8 hours to administer in the paper-based form. And that's a really huge benefit for our participants, because an 8-hour day is a very taxing day for answering these types of difficult questions. And we need them to be enthusiastic to come back in our study. So we do not want to lose the subjects because of the taxing nature of asking these questions. So taking an hour off really has made a big difference in improving our participants' happiness and their retention for longitudinal study. So here's our grand idea. We have all these CRF forms. Well, we really want to be able to convert those CRF forms and those questionnaires about when you were born, your gender, and your disease status, your disease states at those visits, into meaningful knowledge. So we want to convert that into publications and best practices for the clinicians to use in studying the disease. And ideally, we'd really like to inform clinical trials about who and when subjects should be entered into a clinical trial for testing new therapies for Huntington's disease. I already talked about that. So the clinical trials management software that we have currently has 3.57 million records in it from over 4,000 subject visits, from different forms at different times, where all of the data has been converted into a common coding system, and we can perform audits to ensure that it's of the highest quality data. In addition to that, we can take this from our centralized database, and we can share it through the dbGaP system with external collaborators who may have other novel analysis that they'd like to perform on it. And that's it for our public distribution model, is to send it all to dbGaP, where dbGaP helps us to vet new investigators, ensure that they have proper institutional review, and use the data in novel ways that the original grant did not intend this, or did not expect this data to be used in. So in addition to all the clinical data, we have medical imaging data. This is just a quick slide of the brain MRI scans and the subcortical structures and cortical thickness types of analysis that we do. The plot on the left is a diagram starting in 2002 through 2013, the center being in 2002 and the outside being 2013, of every scan session that was created. So each of the little dots in this picture represents data from one of the sites. And as you can see, there are many dots on this plot. Now, each of the pie slices in this plot represents a different site or a different collection site for the data. Additionally, in about 2006, the whole world changed in the types of MRI scanners that were available. Prior to 2006, it was mostly 1.5 Tesla scanners. And after 2006, it was mostly 3 Tesla scanners. And you can see in the middle of the circle all of the orangish and yellowish colored dots. Those are 1.5 Tesla. And in the outer rings, it's mostly blue and dark blue and green dots, which are our 3 Tesla scans. So this diagram does demonstrate the heterogeneity that we have to deal with. Subjects come and go in the study. They don't always live in the same scanner or at the same site. And the data that's collected varies drastically based on site. So for instance, at the top here, you see a couple of sites that have no data in the center rings and have high density of data in the outer rings. Those are sites that came on relatively late in the study, and their data needs to be treated carefully because it represents a time difference between their data and the rest of the study, and we need to make sure that that doesn't bias our end results. The way we deal with our medical imaging data, which is quite different than the CRF data. Medical imaging data ranges from about 100 megabytes per scan session to about 3 gigabytes per scan session. And we currently have over 12,000 images in our repository just for this one Huntington's disease project. So from the different scanners at each of the sites, they are going to send us the data into what we call our pre-archival system. And since this data comes from medical imaging data, we have to be careful that subjects' anonymity is maintained. And so we develop mechanisms so that on the wire, before the data even arrives at our site, it gets de-identified. And the way we do that is to have the sites contact our web server, and then it downloads a little Java applet to their local workstation, and that little Java applet does the de-identification and the data push into our pre-archive. Once it's in the pre-archive, we have trained technicians look at the data, ensure that it's of the proper type, that it has the proper characteristics, and then it goes into our permanent archive. From the permanent archive, we do automated processing and manual processing on that data to generate derived data sets and derived measures. And those again go back, those derived measures and data sets go back into the archive to be organized with the original raw data, and again has visual inspection on the derived data itself, on the derived data that we generated to ensure that again it's of high quality and can be used. Finally, the archive is made available to our external users throughout the world so that they can look at this derived or raw data and save themselves a lot of time in doing quality control checking and in doing pre-processing that is pretty standard across the brain imaging field. This is just a picture of our raw data quality assurance tools. So every scan that arrives at our site is automatically loaded, reviewed for common problems that occur in brain imaging scans, and then cataloged. Then this quality information is cataloged so that we can focus only on the best data that we have for identifying for future analysis. As a secondary benefit of this raw data quality assurance tool is from a multi-site perspective, we review our quality assurance results and identify common problems per site. So for instance, if the signal-to-noise ratio is poor from a particular site, we can contact them and have their MRI scanner technicians verify that the head coils are in good shape and that they are working according to manufacturer specifications. In addition, there's other items here such as whole brain coverage. The protocol states that the subject's entire head and entire brain is to be captured. And we'll often find that at some sites a new technician will be hired and they won't be as careful as they need to be, and we can contact them quickly to have that resolved. That's particularly important with things like brain imaging data because this data costs around $2,000 a piece to collect. And similar things happen for CSF or genetic information where it can be very expensive to capture the data and store the data, and you want to make sure that it's of the highest quality throughout. So our initial data upload from the sites comes in and is organized, and we automatically run a series of tests and create these MRI scan reports that identify just how many data sets are good, how many are bad, and we take this information, these reports, and continuously feed them back to the sites. And it's that feedback mechanism to the sites that has been so helpful for us in improving the quality of our data. By telling the sites where they're failing and how they're failing in a very rapid manner, they can resolve the problems during the course of the study rather than waiting to the end of the study and trying to deal with bad data. And this little plot just shows that in 2004, when we first started giving feedback to the sites, the quality of our data went up drastically between 2004 and 2005. So again, sharing data with other sites. We currently have 48 sub-projects in this major project. We have over 1,900 subjects cataloged and 4,500 independent imaging sessions. Within each of those imaging sessions, we have between 4 and 20 brain image scans of different types. And this is just for the Predict HD project. Doesn't count any of the other projects that I've discussed, or that I mentioned. The Track HD is a foresight where we have 1,700 imaging sessions across 454 subjects. So I'd like to talk just a little bit about the NAMIC family of tools because we work very closely with the National Alliance for Medical Image Computing. I'm currently the president of the Insight Software Consortium, which is one of the foundational software packages for the NAMIC toolkit, the ITK toolkit. This is developed by the National Library, or supported by the National Library of Medicine as a warehouse for segmentation registration algorithms for medical imaging, primarily to support the Visible Human project. And we consume a lot from the NAMIC project, but we also contribute to it. So we contribute to the slicer and the ITK packages, and we wrap the tools that we need in the slicer execution model so that they become an integral part of that data processing system. In order to process our large catalog of data with our large catalog of available tools, we needed to work in a uniform interface. And for that, we adopted this Python-based batch processing system called NiPype. NiPype allows us to use many of the common medical imaging APIs like SPM, FSL, and FreeSurfer with a heterogeneous, that have idiosyncronous and heterogeneous APIs in the NiPype interface, which can provide a very homogeneous API to these tools. And that allows us to mix and match and use different tools for their strengths in one packet, in one platform. And it helps us deal with our scaling issues. So we need to process thousands and thousands of data sets through a very large tool chain, and the NiPype software helps us do that. This is a diagram of our longitudinal data processing pipeline. I understand you can't read this, but what you need to look at are each of those little circles in this processing pipeline is a different software package or a different software tool that is required in order to do this analysis. And the arrows and lines that connect these circles are the inputs necessary to have those tools run properly. And you can see that this is a fairly large workflow with many different moving parts and many different software packages necessary in order to get high quality results. And we need to run this on thousands and thousands of data sets. NiPype is a way for us to manage that complexity, store it, and minimize the amount of work that we need to do. And it is quite a lot of work. So we need to efficiently use our computational resources. This diagram shows the number of CPU days of computation that we've used in the last 2 years. So for this one study, for studying Huntington's disease and processing just the longitudinal data that we have from Huntington's disease, we've consumed approximately 182 compute years. Compute year being one CPU processing nonstop for a whole year. So we've had 182 compute years of data processing done just for the PredictHD project. And one important thing to mention about this is it really does take a whole community effort to get this data processing done. So in addition to computer scientists, we also have graduate students who are processing data on our high-performance clusters, and we have medical students and engineering students and statisticians using this computational infrastructure to process all of this massive amount of data that we have. And it's really important that we have a diverse team of biostatisticians and computer scientists to make sense of this data. Well, of course, 182 compute years is a lot of years. What we would like to do is get the same amount of work done in less time. And so we spend a lot of time investigating ways to be more efficient with the limited resources that we have. And right now we're looking at using add-on cards like the Xeon Phi coprocessor and finding ways to use multi-threaded applications in order to better utilize the compute resources we have. Even though most of our computers that we use have 12 or 14 or 12 or 16 CPU cores in them, we have to make sure that we're using all of those cores efficiently. And in the case of the Xeon Phi, the problem becomes even more difficult because you have 60 plus coprocessors that all need to be used efficiently. It doesn't make sense to use the Phi coprocessor if you're only going to utilize one or two coprocessors at a time. And those are fun, challenging engineering problems that we face every day. Again, the quality assurance tools from our end results of those 182 compute years, we're going to visually inspect them. We have a team of trained experts who go through and look at each of our data sets, making sure that they are of high quality and enumerating where the problems in the data are. And that enumeration is really important for us because that metadata about where the problems are informs us of the types of algorithms that we need to investigate in order to make our data better. So for instance, here we're seeing a lot of susceptibility artifacts in the occipital lobes of our diffusion-weighted imaging data. And that's telling us that we need to consider very carefully the types of diffusion-weighted data that we collect for this study so that in the future studies, diffusion-weighted data can be collected without having susceptibility artifacts in the occipital lobes. And this has been, this quality control checking and massive data analysis really has helped us have cutting-edge results that are informing the community of therapies that should be tried and new research methods that need to be addressed. And this is just one of our very popular papers that show how differences in the prodromal phase, 20 years before external symptoms show up, how we can measure them with this diffusion-weighted imaging data and this processing. And that requires us to make quality assurance tools that include both the measurements and the provenance of those measurements, in our case, in a database. So over the lifespan of this study, over the 14 years of PredictHD, we've actually done many different experiments. And we've had certain data cuts occur, and in 2006 we had our first major data cut created. Well, since 2006, the software tools that we use have gone under extensive improvements. So we need to keep track of how the measurements are made. Even though we've collected caudate volumes for these data sets since the beginning of the study, the way we've collected those caudate measurements has changed over time depending upon the software tools that we used and the advancements in the software tools that we've made. So we have not only the volumetric measurements of the caudate, but the provenance information about the tool, the time that the tool was created, and the type of environment that that tool was run in order to generate that caudate volume. And that allows us to measure over time and have experiments over time and know what caudates are appropriate for combining together for larger longitudinal analysis. Then we can make data reports. After we have that database filled out, we store it in the form that our statisticians like best, which is just an Excel spreadsheet. And for that Excel spreadsheet, we have the data set itself, which are the raw numbers, and we color code those based on the quality scores that we've given them. So the green are the highest quality measures, the yellow are acceptable measures, but we wish they were better, and red are measures that we think should not be used for statistical analysis. And our statisticians can then use that that metadata to produce better statistical analysis. In addition to the data set, we have on our second sheet a data dictionary, and the data dictionary contains the column descriptors, what type of measurement is in that. Is it volume, linear measure, surface area? What biological region did that measure come from? What experiment was used? So what tool set was used for that? What subset of data was used in the analysis? The measurement volume, and then we often will have a URL to a documentation page that better describes that particular measure. And that documentation page either can be Wikipedia or some SNOMED ID that you can go to and find literature related to that particular measure. And finally, this comments allows us to identify every single measure in the database as to its quality and the notes that a rater may have provided on it. So whether the whole brain was covered or whether it had ghosting noise artifact that affected that particular data point. And then analysis tools, we like the R package, but we've been able to work with our statisticians to keep all of our statistical analysis on GitHub, so that we have a historical record not only of the software that we have, but also of the statistical analysis results that we have. And we work closely with our collaborators and our statisticians and our software engineers to have a complete package where we can track across time how these different analyses are changed. And that allows us to access our data, produce the analysis, and ultimately produce the report that we use for understanding the data, all in a software managed control system, and we use GitHub for that. And that allows us to rapidly generate lots of results from many different groups about our Huntington's disease data. And this has really been a key to our success is the software maintenance and the acceleration of neuroimaging, so we can get these papers out from a wide set of collaborators across the United States and across the world on very large, difficult to transfer data. So this is just a second page that shows our public dissemination knowledge. It's not only the publications that we produce in peer-reviewed articles, but also that we use GitHub and public source code repositories extensively in order to share our data. And not share the data, the analysis strategies, and the software used for the analysis. This work has run into many different spin-offs. So in addition to the Predict HD project itself, we also have many ancillary studies and secondary studies from collaborators throughout the world to look at different aspects of Huntington's disease that were not possible to look at within the confines of the original study. And the last thing I want to talk about is just the extensions into NeuroNext. So Huntington's or PREDICT-HD is an observational study, and we're hoping to inform clinical trials to have real impact on these participants. And NeuroNext is a consortium of clinical trials sites that is using similar technologies to coordinate the data collection in clinical trials and efficiently run clinical trials throughout the United States. And just the last slide here is ideas that we have on the future. We'd really like to use external data sources to augment this internal data collection that we have. So we have a tremendous amount of data already. But inspired by the Google Flu Trends work, is there are many different external data sources available as well. So how do we look at the epigenetics of Huntington's disease by taking into account, for instance, environmental factors? And one of the ideas that we have is how do you take these EnviroFacts data service API from the EPA, where you can estimate toxin loads and chemical dispersion facts from coal plants or other types of industrial complexes, and use that information to estimate the toxic effects of those in relation to the diseases that we're studying. So if we can, one of the challenges we're going to have with that is creating these two-way de-identified data sharing, where we can have some type of independent broker that can communicate between different groups so that the data that I have, I can share more easily in a de-identified way, and that other people can share their data with me in a de-identified way, so we can merge those two data sets together and have new findings that were otherwise not possible. So that's the end of my talk. I'd like to thank you for listening and open this up for any questions you may have.
Thanks, Hans. Does anybody have any questions for our speaker?
I have several questions, but I probably limit it just to one or two. Dr. Johnson, thank you very much for a very interesting presentation. And I was wondering whether there are any other data centers that maintain software tools that can be used for other organs instead of just brain. Like, for instance, heart or lung or other, other organs instead.
Yes, there are. The brain has a large community that's worked very well together for a long time, so I think it's got a more vibrant community at the moment. I don't know of a community just for the heart. There is a clearinghouse for brain imaging called NITRIC, the Neuroinformatics Tools and Resources Clearinghouse. I do brain imaging, so I'm not really familiar with heart and lung as well, but I don't know if.
Okay. So is the big data available to the public?
I'm sorry. What was that?
Is the big data that collected through the project available to the public? Like
we are working very hard to make it available to the public. So there's a large portion of data that is available to the public through the dbGaP system, including gene-wide association studies for over 1,000 individuals, and data collected up through, I think, 2008 has been made available to dbGaP. We're in the final year of data collection, and at the end of data collection, we will update that repository to include more data, or to include more results. So it is partially available. It's an ongoing study, so it's not up to date right now. It's a few years behind. But as the study comes to a close, all of the data will be made available through these public repositories.
Hans, I think question that may come to mind for many people who see the presentation is obviously there's a lot of manpower, time, resources devoted to developing this kind of system. Do you have sort of some kind of perspective or words of how to get started if somebody wants to try to start developing some kind of analogous type system? Because many of us are going to have a need to kind of categorize the types of models and the data sets that are generated from the models. We want to be able to share them with collaborators, people that are part of the consortium. We may want to have some checking and model validation, especially in light of the optimization issues you were talking about, if we have students that are picking up code from previous students. So
absolutely. So my experience has been that it's very important to organize your software development process around good software development practices. And the first and most important one is using a source code management tool. Whether it's CVS, SVN, Git, that doesn't matter so much. But you need to have the historical information about what was done, why it was done throughout time in order to have any hope of passing along information from one developer to the other. In particular with graduate students, we've all been there, we've all been bad before. The code that you write, you are so intimately familiar with it because as a graduate student you live and breathe it 16 hours a day for years. When you hand that off to somebody else, they need to be able to know the trials that you went through and the failures that you went through. And the only way that I know to effectively keep track of that is through source code management. And then in addition to source code management, I believe strongly if it's not tested, it's broken. I have seen it over and over and over again that the code works, and it works on the one isotropic standardized data set that everybody in the world uses. And as soon as you give it anything outside of the norm, it just blows up and is completely useless. So testing becomes an integral part. And we really propose that you start testing and write your test before you write your software. Any software development manual that you read tells you to do that. Very few people do. It's hard to get over the first hump because you will take you longer the first time you do it, and it'll take you longer the second time you do it, and the third. But by the fifth time you do your test first and then write your software, you will start gaining advantages and time advantages, and you'll find that it takes much less time to write software that is tested before the software is written. And the problem is that it's hard because the first time you do it, it'll take you a lot longer. And you have to become good and figure out how to write those tests, which is a difficult task. But I am absolutely convinced that it's the right way to do it, and in the long run, it will save you time and money to do it that way. So testing.
If I may ask a question, I have a few, but I'll try to narrow them down. So how much modeling do you do actually with all this data, and where do you start modeling? Actually before recruitment phase, do you model what will results look at the end? What kind of models do you use? Do you, for example, can you predict table 1 that you're going to publish in the paper before you actually start recruiting? Things like that.
Yeah, so modeling is a term that is used in many different ways in the study. Just a second, let me turn my microphone down. So we're modeling the disease process, and so we generate a whole bunch of results in big Excel tables and then fit models to that data that describe how the disease progresses. And using as our input measures things like age and genetic markers, and seeing how those models generate results. And then there's more image processing modeling, So we modeled the shape of the caudate, the shape of the brain, the shape of cortical thickness and gyrus measures, using structural models and using models from finite element analysis and those types of engineering type of models in order to better characterize the biological structure of the brains and the biological changes that are occurring over time.
And I know that Joy has a question in the chat room. I don't know if she can turn her microphone, but unless she asks the questions, you mentioned several platforms that you have to support. Do you use any virtualization technology? VMs?
Absolutely. Absolutely. We use all different types of virtualization because it's cheap. That's the primary reason. We do most of our development on a Unix-based platform, either Linux or Mac, just because that's the way our team has been trained. But many of our end users are actually Windows users. And so I'm not going to expend a lot of money to have a Windows box when I can go to the Azure cloud and have a Windows box created for me. And it's easy to have them created and spin them up and shut them down as necessary. So yes, we use a lot of virtualization software for that testing platform. And that allows us to, within our lab structure, we have a fairly stable environment that we work in day to day, which translates to fairly old. You know, it's not bleeding edge versions of Linux or anything like that. But that way we have a nice stable platform. But when we want to test whether our software is going to have legs in the future, we need to be testing on the latest GCC 4.9 compilers with the latest optimization capabilities. And so we will just spin up virtual machines and test in those. And when we're done testing, just shut those virtual machines down.
Joy, did you have a comment?
Yeah, Hans, thanks for the great presentation. I think it's really impressive, the testing that infrastructure you've set up with CDash. My question was, how much resource do you put in, both computing resources as well as manpower, to keep your CDash infrastructure up and running? And do you have any thoughts on, if you have another small group or an individual who wants to set up a similar type of testing infrastructure, what does it take to kind of help them over that hurdle to get them up and started really quickly?
So first off, I would recommend using publicly available resources if you can. We used to run our own CDash system in a virtual environment, and then I think it's called CDash.org came available, which is publicly available. You can just register your project, and as long as you don't need your project to be hidden and private, you can just use publicly available resources. So if your testing isn't proprietary or need to be kept secret, I would recommend that you use publicly available resource. Other than that, if you need to have your own version, they do have some virtual machines that you can download the whole VM and start that up. That's not too hard. And they're just standard LAMP-based web instances, so there's nothing really special about it. You need to have a good system administrator or a good system-savvy graduate student spend 6 hours figuring out how to set it up. As far as source code management, I'm a big fan of GitHub and Git, but that's because I work on projects that have thousands of developers. Git does have a very large learning curve, but it's incredibly powerful. I mean, it's incredibly powerful. So if you work on big projects, you will eventually have to learn and love Git. If you're working on a small project with just 3 or 4 people on your team, you know, maybe set up your own SVN server. That might get you by for a while. As far as time, these things do take time to set up. I am convinced that you will reap rewards by spending this time, though, because I don't know. Every month, somebody comes to me and asks if I can help them identify a bug in their software. And inevitably, they send me a tarball that they pulled off their graduate student's computer that's got no history, no documentation, no test data. And, they said, this is a fabulous lung imaging registration, and we've used it for years, and we have the binary, but the binary doesn't work on our new system. But they don't have any test data. They don't have any reference data. I can't even tell whether I get the right results if I put the data in. And so it's really hard to start from that and identify what changes can be made safely. So having a great test suite for your software is very valuable for making sure that your software lives beyond the one data set it was used to develop the hardware or the software.
Thanks, Han. Actually, so to build on that, we have the simtk.org infrastructure where we have Subversion repositories set up that people can get started on quickly. But listening to what you've been saying, I'm wondering, are there incentives that we might build in to try to get people to follow a more rigorous software development protocol?
Yeah, it depends upon your community. So in the ITK community, we spend a lot of time cultivating our community, providing hands-on sessions of how to properly develop software, and giving feedback to users that are new to the group so that they understand the benefits of developing software in this manner. And that cultivating that community is hard work. It does take time, but the benefits of it, you get to reuse the software from different teams, and it really does allow for collaboration. I think that without having that type of desire to cultivate the community and cultivate a common set of standards, you cannot effectively share software between different groups. It doesn't work out very well.
Thanks.
No problem. Any other questions? I'm sorry, I can't hear you. Grace?
Oh, sorry. I should mention that both of these efforts that Hans has mentioned and Joy has mentioned are stemmed from the National Centers for Biomedical Computing supported by the National Institutes of Health.
I see.
Well, thank you very much for this opportunity. I appreciate it.
I have a very trivial question.
Sure.
In terms of publications, so whoever use that big data, do they have to include all the PIs, co-PIs on any publications? I'm just curious.
So you know that the answer to that isn't trivial and easy. It's very, very, very difficult. It depends upon the project. With the Predict HD project, we did spend a lot of time, a lot of painful time discussing the pros and cons of different approaches. We do have a global the Huntington's disease research group that we ask be included as an author, but we do not require all 200 collaborators to be listed.
Okay.
It's just one global name. All right. Any other questions?
I have another. If you have time, I have another question.
Okay.
I think it's very important to have this kind of data center to maintain the software and then standardize the data quality. And recently, I saw several commercial companies became this kind of practice. That means they are not willing to release their source code or their not source code, their product to researchers. Instead, they ask researchers to send data to their company for analysis. And so gradually, this company collect large data set. Maybe these data sets are sponsored by NIH, but then somehow they kind of collect this data. So they decide who can use this data. So in your opinion, is there any way to kind of address this issue?
How would you want to address it? What's wrong with that?
Well, then that means if that company is not willing to say kind of release the software to this group, then that group probably cannot do research in this area. And they also start collecting data from different research groups because they are not going to release their software to research group. Instead, they ask a research group to send data to the company. So in fact, the company kind of collecting big data.
Yeah.
And that big data is not kind of available to the public.
You know, I don't think that I see a problem with that.
Okay.
That's yes, that does happen, but there can be good that comes from that as well. And the company may not have a financial incentive to put forth the effort to make that data public. And if they don't, then I don't know that we can mandate that. I suppose you can make it so your data isn't available, but I think that has cons as well. I know that this is I'm coming from the Insight Software Consortium, where the software that we develop there is explicitly designed for companies to take, commercialize, and not reshare. We want them to take the software and the efforts that we put in as a community and benefit from it. Because it's our belief that that will provide for a rich environment where more tools are available. Even though those tools may not be free, those tools otherwise wouldn't even exist.
But how about data, like imaging data? Because imaging data is very expensive, right?
Yeah.
So if say some private company have large collection of imaging data and they kind of perform their own analysis without kind of make it available to the public, and if this data are supported by NIH through different grants, then I feel that this data should be available to any research groups instead of the company. Anyway, it's kind of different.
I think that's an issue that needs to be taken up with NIH policies, and I don't know. I guess I don't see any clear and obvious solution to that or problem with that.
So the NIH is trying very hard to address your questions, Jinglong.
I see.
And we have the new Associate Director for Data Science, Phil Bourne, who joined us, and he's being very active in trying to address these questions.
Okay.
It'd be interesting to see if you guys want to talk more about data sharing. You can post it in the wiki as an open discussion.
Okay.
If you want to have some discussions during the September meeting too, just feel free to suggest it.
Okay, I will. Thank you.
If you have a few more minutes, I will ask one last question. You showed a slide that shows the improvement of your operation. Can you quantify it, and will you attribute it to technology or to something else, this improvement?
Oh, yes, we can quantify it. Now the question is whether our quantification methods, if you agree with them or not. We did with a series of measures that we thought were important for medical imaging, looking at factors that we know have impact on the results, and then created a metric from that. So, for instance, our protocol required that this voxel sizes be 1 millimeter voxels, and so if they are not 1 millimeter voxels, we gave that a penalty, and the further you deviated from 1 millimeter voxels, the more penalty we ascribed to that. And it can't be the technology, because the hardware and the technology didn't change. What changed was the site's conformance to the protocol that we provided.
You're saying it's usable, the using of the fact that people use the technology better. This is what actually made things better.
The fact that they followed the protocol. Our protocol stated that it was supposed to be, for instance, 1 millimeter isotropic voxels. And our reporting software could identify when they sent us data that was not collected properly. And by giving that feedback information back to them, they could adjust for that.
Okay.
And then, that's the simplest example, but we also did measures of noise characteristics in the image, and when we saw that their scanners started to drift and have a higher level of noise, we could again call them and ask their technician to recalibrate their scanner.
Thank you.
All right. If there aren't any other questions or comments, let's give a virtual hand to our speaker.
Thank you.
Thank you, Hans.
Yes.
Thank you.
Have a good afternoon.
You
Bye-bye.
too. Thanks a lot.
Thank you.